In a previous video, I suggested that the whole person reorganizes around a survival-oriented state consequent to trauma. In this video, we'll explore how the body reorganizes around trauma, focusing on specific systems. These systems are included in the holistic evaluation and treatment of trauma from an integrative psychiatry perspective. Because trauma and stress involve a high energy requirement survival state, the body attempts to mobilize energy stores to sustain this. This is done through the release of cortisol, which mobilizes energy stores. Normally, cortisol levels rise until feedback mechanisms in the body tell the adrenal gland that the threat is under control, and the levels fall back to normal. In the context of trauma, this feedback mechanism becomes disrupted, and cortisol continues to spill into the bloodstream. Over time, this exhausts the adrenal gland's capacity to keep up, and the individual shifts from a hyperadrenal state to a hypoadrenal state, sometimes called adrenal fatigue. Cortisol has several detrimental effects when it's not adequately managed. First, it disrupts the metabolic system by altering how we metabolize carbohydrates, fats, and proteins. Diabetes and abnormal fat deposition are longer-term consequences of this. Second, it interferes with the normal synthesis of neurotransmitters. For example, cortisol activates the kynurenine pathway, which steals tryptophan, a normal precursor to serotonin, and converts it into toxic concentrations of kynurenic acid, which increases glutamate, an excitotoxic neurochemical, when found at higher levels in the brain. Outside of serotonin, cortisol can interfere with the activities of dopamine and norepinephrine. Third, cortisol directly and indirectly contributes to a pro-inflammatory state by activating inflammatory cytokines through the stimulation of macrophages and microglia. This pro-inflammatory state compounds the changes we've already mentioned, amplifying metabolic and neurotransmitter disruption. Fourth, cortisol is integrally related to the circadian rhythm. Not only is this responsible for maintaining normal sleep-wake patterns, but it's also related to the normal ebb and flow of neurochemistry. In the brain, cortisol abnormalities interfere with the activities of the prefrontal cortex and hippocampus, impairing executive functioning, memory, emotion regulation, and top-down control of impulses, among other things. Through advanced laboratory and diagnostic testing, the activity of the hypothalamic pituitary adrenal, or HPA axis, can be evaluated in detail, clarifying the activities of cortisol. The HPA axis and the metabolism of cortisol make up half of the larger steroidogenesis pathway. The other part of this is the hypothalamic pituitary gonadal, or HPG axis. This axis includes the activities of estrogen, progesterone, and testosterone and their breakdown products. By nature of the interrelationship between the HPA and HPG axes as part of the steroidogenesis pathway, some speculate that when one is influenced, the other is consequently influenced. With this being the case, individual hormones demonstrate a complex interrelationship with other hormones. This interrelationship becomes part of the more overt expression through clinical signs and symptoms. Estrogen affects the prefrontal cortex, hippocampus, amygdala, hypothalamus, and locations in the midbrain responsible for the synthesis of monoamines, namely the Raphe nucleus and the locus coeruleus. A healthy balance of estrogen supports the biosynthesis of serotonin, dopamine, and norepinephrine. The selective serotonin reuptake inhibitor antidepressants are more effective in the presence of healthy estrogen concentrations. However, estrogen, like all other hormones, must go through a breakdown process through liver detoxification pathways. An accumulation of specific estrogen metabolites, E1 in particular, can be problematic. Accumulations compete for binding on certain estrogen receptors in brain regions that have been determined to be anxiogenic, generating anxiety. This outcompetes the activities of anxiolytic estrogen receptors. These estrogen metabolites are also inflammatory because they stimulate the kynurenine pathway in microglia to shunt tryptophan into kynurenine metabolites, leading to a pro-inflammatory and excitotoxic state. Progesterone has a protective effect, supporting the activity of E2, the beneficial estrogen metabolite. This shifts the competition, favoring binding to anxiolytic estrogen receptors. Outside of that, progesterone has been shown to be generally neuroprotective. Hence, its increasing recognition as an important consideration in perimenopausal support to reduce the likelihood of neurocognitive decline. Testosterone metabolites, when in high concentrations, contribute to anger and irritability. On the other hand, when deficient, this tends to contribute to a more depressive experience. Testosterone metabolism is highly contingent upon the state of the internal environment, where a pro-inflammatory internal environment tends to generate more toxic testosterone metabolites. A full evaluation of the HPG axis can be done, illuminating the interrelationships among these hormones. The enteric nervous system in the gut can be considered the second brain of the body. The neurochemistry in this system mirrors what we see in the central nervous system. In fact, it's responsible for the synthesis of 90% of the body's serotonin and accounts for 70 to 80% of the immune system. Stress and associated HPA axis activation lead to impaired intestinal integrity through dynamic interactions with the enteric nervous system. This change in integrity increases the susceptibility to dysbiotic environments, where there's an imbalance of commensal or beneficial versus opportunistic organisms living in the gut. This shift in diversity and abundance generates a host of systemic disturbances. One of the most noteworthy disturbances is its influence on the immune system. Bacterial antigens and metabolites translocate into the system through this more porous intestinal barrier, triggering an immune reaction. This immune reaction includes the generation of several pro-inflammatory cytokines, which cross the blood-brain barrier and lead to neuroinflammation. Under the influence of neuroinflammation, monoamine activities are disrupted, and larger networking issues between brain regions are realized. This process is nonspecific and can contribute to depression, anxiety, and executive impairment, or ADHD, among other symptoms. What's more, this is only one subpathway. A disruption in the intestinal microbiota significantly influences the body's ability to pull critical micronutrients out of the gut to be used in several metabolic processes, including those crucial for neuropsychiatric activities. This is compounded by the influence this dysregulation has on insulin sensitivity, glucose metabolism, and lipid balance. These imbalances increase the risk for metabolic syndrome, obesity, and neurodegenerative changes, often comorbid occurrences. Through advanced laboratory and diagnostic testing, the gut can be analyzed in detail, giving a glimpse of these activities and where to target treatment to bring more balance. In the context of HPA axis activation and the resulting cortisol elevation in trauma and stress, the hypothalamic pituitary thyroid, or HPT, axis is suppressed. Specifically, thyrotropin-releasing and thyroid-stimulating hormone are both suppressed. The downstream effect of this is lower thyroid hormone output, specifically T3, the active hormone. Stress reduces the activity of deiodinase enzymes responsible for the conversion of T4 to T3. It sometimes increases the production of reverse T3, an inactive form of T3 that blocks T3 receptors. Because stress and trauma can disrupt immune tolerance, there's an increased risk of autoimmune thyroid disease, especially Hashimoto's thyroiditis. Stress affects estrogen, progesterone, and testosterone, which all influence thyroid function and receptor sensitivity. Finally, stress depletes selenium, zinc, iron, and iodine, all critical for thyroid hormone synthesis. These are all changes that occur in response to stress, directly stemming from the impact trauma has on the HPA axis. Note that there is a compounded effect through the interrelationship among the systems already discussed. Subclinical thyroid dysfunction from stress often manifests as symptoms that overlap with psychiatric conditions, leading to an under-recognition of thyroid involvement. Stress and trauma can mask, mimic, or magnify thyroid dysfunction, creating both direct hormonal disruption and a cascade of immune and metabolic imbalances that affect the entire body-mind system. A full thyroid axis evaluation should include a TSH, T4, T3, and reverse T3. Additional markers such as antibodies should be considered in the context of these findings. Along the way, we've explored several links to the immune system. Let's look at this system in a little more detail. Acute stress temporarily boosts immunity as part of the fight or flight response. Chronic stress, on the other hand, leads to prolonged cortisol elevation, which initially suppresses immune functioning through a reduction in lymphocyte and natural killer cells. Over time, this causes a glucocorticoid resistance, where immune cells stop responding to cortisol's anti-inflammatory signals. This results in chronic, low-grade inflammation. Trauma tilts the immune system toward innate or inflammatory dominance and away from adaptive regulation. This is reflected through an increase in pro-inflammatory cytokines such as interleukin-6, tumor necrosis factor alpha, and interleukin-1 beta, and decreased T regulatory or Treg cells, impairing immune tolerance. This imbalance contributes to conditions like autoimmunity, allergies, and chronic infections. In the brain, the kynurenine pathway, governed by microglia, is activated by inflammation, cortisol, and estrone. When activated, tryptophan is converted into kynurenine and ultimately into two byproducts, kynurenic and quinolinic acid. These two byproducts, when imbalanced, lead to disruptions in dopamine, norepinephrine, and serotonin activity, and increased glutamate. The ultimate consequence of this on a larger scale is a picture of neurotoxicity and reduced neuroplasticity and synaptogenesis, magnifying into larger neural networking problems. Early life trauma, as evaluated by the 𝐸𝐴𝐳s, can cause long-lasting epigenetic changes to immune-related genes. This reprograms immune responses to be hyperreactive or ineffective, increasing susceptibility to autoimmune disease, infections, and psychiatric syndromes reflective of neuroinflammation. The brain and immune system communicate through the vagus nerve, cytokines, and neurotransmitters. By inducing a hypervigilant immune state, physical and psychiatric flare-ups are more frequently triggered. The activities of the immune system can be evaluated through advanced labs and diagnostics, looking for signs of baseline inflammation, autoimmunity, and interactions from other interrelated systems. Trauma and stress can significantly deplete or dysregulate micronutrient levels in the body. These subtle shifts in vitamins and minerals have wide-reaching effects on mood, immunity, energy, detoxification, and repair. Chronic activation of the stress response ramps up metabolism and detoxification. This increases the demand for key nutrients used in neurotransmitter synthesis, antioxidant defense, and hormone metabolism. Some of the most affected include B complex, magnesium, vitamin C, zinc, and selenium. Some of these, as we've touched on, Some of these, as we've touched on, are implicated in thyroid functioning. Stress increases reactive oxygen species generation and inflammation. Subsequently, antioxidant micronutrients like vitamin C, E, glutathione precursors, and selenium are rapidly depleted to counteract this. This imbalance promotes cellular damage, neuroinflammation, and immune dysfunction. Because trauma affects the gut via the gut-brain axis, dysbiosis and impaired intestinal permeability reduce nutrient absorption. What's more, digestive enzymes and stomach acid, required for the absorption of iron, B12, and magnesium, among others, decreases under the influence of chronic stress. The result is nutrient deficiencies even with adequate intake. Stress-induced behaviors like poor eating and substance use lead to the loss of magnesium, calcium, potassium, and water-soluble vitamins like B complex and vitamin C. There's also a vicious cycle here, where micronutrient deficiencies can exacerbate symptoms of trauma and stress. For example, low magnesium can contribute to more anxiety, insomnia, and hypervigilance. Low B complex impairs methylation and contributes to neurotransmitter imbalances. Low zinc impairs immune function and increases inflammation. And low vitamin D impairs the immune system and contributes to mood dysregulation. In this way, trauma and chronic stress act like a nutrient wildfire, burning through reserves, disrupting absorption, and preventing replenishment, leaving the body vulnerable, dysregulated, and less able to heal. These micronutrient changes can be fully evaluated and treated through advanced laboratory and diagnostic testing to bring about more balance and support the system as a whole. Trauma, especially chronic and early life trauma, can trigger epigenetic changes that influence how genes are expressed without altering the DNA sequence itself. These changes have long-term effects on stress reactivity, immune functioning, mood regulation, and even intergenerational health. Through the activation of the HPA axis, trauma floods the body with cortisol and stress-related neurochemicals. These signals lead to epigenetic modifications, especially in DNA methylation, histone modification, and non-coding RNA expression, all influencing the way genes are expressed. Through these activities, trauma silences or amplifies key genes involved in the HPA axis, immune system, neurotransmitter, and oxytocin systems, all resulting in stress sensitivity, persistent inflammation, immune dysregulation, mood instability, and difficulty with bonding and emotion regulation. Childhood trauma, often measured through the 𝐸𝐴CE questionnaire, occurs during critical developmental windows. The brain and epigenome are more plastic. Trauma can quote-unquote set the stress responses for life. This can lead to overactive or numbed stress responses and an increase in the evolution of neuropsychiatric syndromes, autoimmune, and metabolic disease, among other outcomes. Trauma-induced epigenetic changes can be passed along through germline cells or through maternal stress in utero. Children of trauma survivors may carry biological sensitivity to stress even without direct exposure. This is observed in descendants of Holocaust survivors, war veterans, and population with systemic trauma. Trauma is not just a psychological event. It writes itself into the body through biochemical tags that tell your genes how to behave. This helps the body survive danger, but at a cost. It may stay stuck in survival mode. While trauma leaves a molecular footprint, it's not destiny. Growing work in the field of epigenetics has provided clinical tools to evaluate a variety of these genetic changes and reorganize these processes through treatment and support. A holistic approach can reshape gene expression over time. Trauma is not just a memory of something that happened in the past. It's a biological event that reshapes how the body functions in the present. Its effects ripple across multiple systems, creating a body that remains in survival mode, even long after the danger has passed. As I touched on in a previous video, trauma is not in the event, it's in the response. It's in your cells, nervous system, immune responses, and biochemical wiring. But it's also malleable. With safety, connection, and targeted support, these systems can begin to recalibrate, heal, and restore balance. If this perspective resonates with you, and you want to see more content, please like and subscribe. Leave a comment if you would like for me to cover a specific aspect of trauma that I haven't touched on yet, and stay tuned as we continue to dive even deeper into the nuances of trauma. To learn more, check out the additional videos and links in the description below.